All right, we're going to get started tonight. Tonight, of course, there's the exam review. If you don't remember, it's going to be in GN 101, 6:30, till we finish, till done. Like last time, it's supposed to be done at 8:30, but we ran over by like 30 minutes last time. Same thing this time. We'll do 6:30 till we're done. I'll try to keep it within 2 hours, and we'll see what happens. Your exam is next Monday, the 15th. Right. Same thing, make sure you bring pencils, scantrons, all that stuff. There's always at least one person that forgets something, so please try to bring all the required materials. You will, again, like previously, as long as no one's using the room afterwards, you may be given an extra 5 or 10 minutes than the required 50 minutes. And it's going to be somewhere between 30 to 35 questions long. I'll go over potential extra credit questions tonight as well, so stay tuned for that. There will, of course, be one extra credit question, and just like for exam 2, as long as you explain it enough that you have proven to me that you know what you're talking about, then you'll be fine. All right. Oh yeah, announcement. 29B will not be on exam 3, so I bet a lot of you are relieved about that. But that does mean it will be on the final. That means for exam 3, you need 10A, 10B, 11, 25, and 29A. So you need to know these PowerPoints, these chapters for exam 3. Oh, 28. Okay, thank you. Plot twist. Okay. Not for exam 3, no. This is all you need for exam 3. 10A, 10B, 11, 28, and 29A. For the final, obviously you need everything we've learned, but the final will also have all the chapters we've learned in addition to 29B, 30A, and 30B. Okay, so those will be covered. 29B has already been covered some, of course, but 30A and 30B, I'm actually going to start on Thursday's SI session because we got to kind of go a little bit ahead because I want some of the last few SI sessions to be reviewing stuff from many, many weeks ago from exam 1 and exam 2. So I'm going to go a bit ahead. Thursday's SI session, I'm going to start on 30A, and next Monday's SI session, I'll have it be like, last second questions for people. And then next Wednesday will be 30B, and then the rest of the SI sessions will be reviewing for the final. Okay, so that's the plan. All right, so today, though, I'm going to outline some things, not in as much detail, of course, as what you're going to get tonight, but how to study for this exam and for the final. I've said it many times, you should be outlining your notes in different pages, so I'm going to have you guys tell me what to write down for certain things, all right? So, if I want to review 10A and 10B. I know 10A talks about nitrogenous bases, nucleotides, nucleosides, some special nucleosides, and things like that. So let's see, what are some things you guys remember? Let's start from the simplest thing, and that is nitrogenous bases. So let me write that out here. I won't be fitting it on one page here, but a good practice for you guys is if you want to test yourself to see if you remember everything, all those important concepts, try to write them all down from memory. It sounds harder than it actually is, but try to outline things from just purely from your memory. Then you go check your notes to see if you've forgotten anything or gotten anything wrong. The more efficient you can do this, the better that you will do on the exam. So, nitrogenous bases is the very first thing that they talk about. There are two categories, which are what? Good. Purines and pyrimidines. And you can give yourself some nice shortcuts to memorize how they look. Purines will have what general feature? A double ring, right? Two aromatic rings. All right, pyrimidines, single one. Okay. What are the nitrogenous bases that are classified as purines? A's and G's are your purines. What about pyrimidines? It's the other ones, right? C, U, and T. Okay. So all from memory is what you should be practicing. Be able to write out these notes like this. Let's start with the pyrimidines here. If you were to identify one structure from another, you would look for really only little differences between them. You don't have to memorize every single carbon and nitrogen in these things. I personally, and you guys might have different ways to memorize these things, I personally memorize cytosine or as having an amino, whoops, that's a double bond there, as having an amine group at the top there. All right. Uracil will instead have a carbonyl there. And thymine, this is just how I memorize it, has a carbonyl with this methyl group tail attached there. Obviously, there's more things that I haven't drawn in each of these components, but if you're just trying to tell one from another, this is usually enough. So again, cytosine, there's an amino. Uracil, there's a carbonyl. Thymine, there's a carbonyl plus a methyl group there. Adenosine, or adenine, I'm sorry, adenine. What functional group, and again, it might be different from what you guys memorize, but what functional group does adenine have that guanine doesn't have as many of? An amino group. Adenine's going to have an amino group there. All right, and in fact, it has a couple amino groups, but if you look there, it's going to be an amino group. If you look at that exact spot, though, on guanine, what do you have instead? A carbonyl. Okay. There's other stuff I haven't drawn on those, but whatever. It's a multiple choice exam. You just need to know them just enough to pick one from the others. All right, then these are the general features that I look at. They're all aromatic rings, so there's some properties associated with your nitrogenous bases. One of the easier ones to initially memorize is if they all have aromatic rings, what can they absorb? UV light, all right, good. Let's see. They all absorb UV light. Then there's two other stuff. Well, they're all nitrogenous bases, so they can participate in acid-base reactions. That makes sense. Then there's that one phrase that you hear in organic chemistry that we don't really talk about too much in this class, but if you see the phrase, just be able to recognize it. Keto-Enol Tautomerization, something. Keto-Enol whatever, that is a property of your nitrogenous bases. Basically, an oversimplification of what this means is that carbonyl groups can flip into hydroxyl groups and back and forth. Certain combinations of that. I'm not going to draw it out, but they're illustrating that all of your nitrogenous bases have the capability to undergo keto-enol tautomerization. Really, out of this feature, the important part is that the keto form is the preferred form of your nitrogenous bases. It's your more stable form. For this class, that's what you need to know. Technically, if you look at it deeper, that is not true for all your nitrogenous bases, but for this class, we simplify it and say the keto form is the preferred form. All right? Well, there's probably more with nitrogenous bases. Somewhere in the notes they talk about your RNA bases. I'm not even going to bother writing those down because you aren't required to memorize special RNA bases. These are your core ones. There are some funny names with them like xanthine, hypoxanthine, blah, 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 that stuff, whatever. So it's not very important. After all this, we're going to start making it a little bit more complicated. is when we start attaching nitrogenous bases to sugars. So let me move this up a bit. Those are nucleosides, right? Still recording, right? All right. Your nucleosides, which are your nitrogenous bases plus one of two options, ribose, or later on you're going to see deoxyribose. All right? So whenever you have a nitrogenous base attached to a sugar and nothing else, no phosphates, I should say. If there's no phosphates involved yet, it is a nucleoside. The names of your nucleosides, the names of your nitrogenous bases change. The suffixes change depending on which nitrogenous base that you're looking at. Adenine turns into adenosine. Guanine turns into guanosine. Right? Your pyrimidines instead end like so. Cytidine, uridine, and thymidine. All right, so if you see a picture of a nucleoside, would you be able to label the correct name? Pretty simple. Out of all these, does anyone happen to remember which one is the only one with biological activity? Adenosine. This is the thing that makes you sleepy, right? And part of the reason is there are things called adenosine receptors. Fatigue and sleepiness arises when adenosine binds to those receptors. Caffeine, it turns out, blocks those receptors. So that's one of the many ways caffeine increases alertness and reduces drowsiness. But adenosine is the only one that at the nucleoside level has bioactivity. We learned about other nucleosides, though, that have bioactivity, but these are those artificially synthesized stuff, right? And you don't have to know the exact name or how to spell them exactly right, but if you see them as an answer choice, you should recognize what they do. One is something like azacitidine. Anyone remember what this does?
Shuts down the enzyme.
What's that?
Shuts down the enzyme.
So it shuts down a particular enzyme that is associated with what disease? Cancer. It is associated with a disease called acute myelogenous leukemia. This drug inhibits the enzyme that is somewhat responsible for that. And you're going to have to look at your notes for more details. I'm going to oversimplify it. Azacitidine inhibits an enzyme, and maybe I even have this backwards, but that's for you guys to go look at the details. I believe it inhibits an enzyme that removes methyl groups. Inhibits enzyme that removes methyl groups. So again, disclaimer to anyone listening on the video or those that are here right now, go check if this is right or wrong. These are some very specific details that I don't usually remember too well, but I'm not taking the exam. So azacitidine inhibits the enzyme that removes methyl groups on chromatin. All right, the specific details are what you need for multiple choice answers. The general memorization stuff is what I'm going to be writing down on here. The general features, you can fill in the details on your own. That'll help you study. So, some drug helps inhibit some enzyme because that enzyme is linked to a particular cancer called acute myelogenous leukemia. All right, and this disease is associated with over-methylation of a particular, again, look up the details, but is associated with some pattern of methylation at a particular site. Yeah. The enzyme or the drug does? The drug, okay. So it causes hypomethylation, so it inhibits enzyme that removes. So I have that double backwards here. So yeah, all right, I'm just going to erase this, and we're just going to write down hypomethylation. Something does hypomethylation. The drug causes hypomethylation, right? Not the enzyme. Okay. See? Easy for me to forget that because it's like double negatives here. All right, so this causes a reduced methylation at a particular spot. The end result is this helps treat a particular cancer that is associated with overmethylation at that particular spot in the genome. I'm trying to think if there's anything else with that drug. Yes? It sounds like, and this is going by your peers, that azacitidine affects methylation of a particular chromatin site. Is that right? Yeah. Anyone else confirm that? Okay. And it's hypomethylation that the drug does, right? Which means less methylation, right? It inhibits an enzyme that normally causes methylation. So the end result is there's less methylation. Okay? All right, so that's azacitidine. Again, don't worry too much. It'll be one question or maybe even just one answer choice in a question. So if you're trying to memorize every single mechanism azacitidine is responsible for, don't stress too much. There's some other ones, of which I'm not usually going to memorize. Something about targeting a virus, right? Targeting some virus, thymidine kinase. I don't know if they even give you the name of that drug. If so, make sure you are able to recognize the name of that. I personally don't remember it, but you should look at whatever the name of that is. This is good practice. I'm giving you just bare bones stuff, and you guys should be filling in the details. Basically, this nucleoside drug, the way it works is very specifically targeting the virus version of an enzyme, and it leaves alone the human version of thymidine kinase. So it's a very nice drug. The only other one I remember, and there might be more, the only other one I remember is ICAR, because this one's a cool one. Anyone remember what ICAR increases the production of? Mitochondria. Very good. That they use in what? I don't know that much detail. Maybe. If it says it on there, stick with that. But you know, that's up to you guys. You guys have to write these things out. You have to outline your notes. And the more that you can do so from memory, the better you're going to do. And you might find that you'll probably do better than me on exam 3 if I had to do this. But there you go. These are nucleoside drugs. There might be more than this, but these are the ones that I can remember sort of just off the top of my head. ICAR is a mitochondrial production factor. And, you know, for those starting to worry that, you know, I'm not giving you everything, or during the exam review, I will give you stuff in more detail, of course. But for now, this is just getting you guys to practice outlining things on your own. All right. After nucleosides, we have nucleotides. Nucleotides are very simple. It's just nucleosides plus what extra thing? Phosphates, right? So sides plus phosphates. And depending on how many phosphates you have, they usually change the name from adenosine to adenosine monophosphate, adenosine diphosphate, and adenosine triphosphate, blah, blah, blah. So in this section of the notes, you should be able to identify a particular nucleotide if the structure was given to you. I'm not going to draw anything here. But other things, such as functions, you should know what ATP is responsible for, GTP is responsible for, UTP and CTP are each responsible for. ATP should be the easy one. This one is metabolism. GTP you'll learn about more in detail in chapter 30. What is that one involved in? Anyone happen to remember? Protein synthesis. It is translation. This thing is necessary for translation. And I always get these two mixed up. One of them is lipids and the other one's carbohydrates. Did anyone remember? CTP is lipids and this one is carbs. Okay. And I think UTP is carbohydrate metabolism, but CTP is lipid. Is it synthesis or breakdown? Synthesis? Okay. See, so fill that in. I believe UTP, if you take Biochem 2, if you're studying for the MCAT, UTP is involved in both the building of glycogen and the breakdown of glycogen. Okay. Let's see. There's another one. There's another form of nucleotides called cyclic nucleotides. One example you might recognize is cyclic AMP, cyclic adenosine monophosphate, which is a signaling molecule. Back in exam 2, you don't need it for this exam, but back in exam 2, you might remember that cyclic AMP is involved in the protein kinase pathway. For this exam, the only other place we've seen cyclic AMP is it has some relationship with something in prokaryotes, right? With the lac operon, right? We'll get to that later. But cyclic nucleotides are involved in signaling. Is there anything else with nucleotides before we start putting them together? Anyone happen to remember? I literally don't remember, so if anyone remembers, I'm fine with writing it down here. All right. Let's see. After this, we have nucleic acid, so we're building it up. Okay. And yes. Just that if you see cyclic anything, it's a different signaling molecule. So yeah, cGMP is its own thing. Nucleic acids, you have RNA and DNA, of course. They differ simply on the sugar and the presence or absence of a single base, right? RNA has uracil, DNA has thymine. They both have everything else. RNA is made of ribose. DNA is made of deoxyribose. All right. When we build nucleic acids, we always build them in a 5' to 3' direction. That's true for both of these. We always build them in a 5' to 3' direction. The reason why, I'm not going to draw this either. That is up for you guys to practice and identify. The reason why is that on the 3' end of every single nucleotide is a hydroxyl group. The hydroxyl group is what you're going to use to add more stuff to it. On the 5' end, what functional group do you have instead? A phosphate. So there's a phosphate at the 5' end, and there's a hydroxyl group at the 3' end. The phosphate of the next nucleotide, the 5' end of the next nucleotide, will react with the hydroxyl end of the forward nucleotide. And so that's how we build nucleic acids, and that's how enzymes that you'll learn in chapter 28 and 29 build nucleic acids. I don't know if it's in this section or not, but there are enzymes that exist to cut nucleotides. So forgive me if I'm going a little bit out of order, but I just happen to remember it here. I'm going to do that right now. Let's see. And I'm specifically talking about exonucleases. So if this doesn't belong in 10A or 10B, you can put it where it belongs to, but this is what I'm remembering. And again, you're building it from memory. The more you can put down, the better you're doing. So exonucleases, I'm going to make up a sequence. A very common exam question will be you're given a sequence and maybe you use a 5'A exonuclease. What is the result of doing so? Well, let's see. So what an exonuclease does is that it removes nucleotides one at a time from the exterior ends of a DNA or RNA sequence. You can contrast that with an endonuclease, which will cut somewhere in the middle of a DNA or RNA sequence. So exonucleases are only able to remove one at a time nucleotides from the ends of a sequence. The specific name attached to the exonuclease gives you a hint at how it will do this. The 5' part in the name indicates that the exonuclease will remove a nucleotide on the 5' end of that nucleic acid. So in this case, in problem number 1 here, this exonuclease will try to remove cytosine from the 5' end. The A part tells you how it will do that. So I'm going to draw the first couple bases here. Oops, that's a C. All right, so I've drawn all those phosphates, phosphate backbone of this nucleic acid. A 5' A cut, an A cutter, by definition, will cut before a phosphate. So it'll try to make a cut here, which is before a phosphate, but if you cut there, nothing happens, right? Nothing has changed. It'll then try to make a cut right here, because that's before this phosphate. And in doing so, it will remove the first nucleotide here. So you are left with that fragment and this fragment. And there will be an answer choice that will look like this. These are the fragments you will make if you use a 5' A exonuclease on this sequence. Feel free to practice any other combination of exonucleases. 5' B exonuclease, 3' A exonuclease, 5' B exonuclease. There are 4 possible exonucleases that you could apply here. I'm not going to go over the other 3 here. All right? But the 2 features you're looking for is this number tells you what end of the nucleotide you are going to cut, and this one tells you what end of the phosphate you're going to cut. A cuts are always before a phosphate, B cuts are always after a phosphate. All right? So let's see, nucleic acids. Again, I'm probably forgetting some things, so I'm probably just going to move on to 10B for now, where we talk about polycistronic and monocistronic stuff. I'm sure in 10A there's a brief slide that talks about the different types of RNA. So make sure you memorize that slide. I mean, a lot of that you should know you should have known since bio, which is like mRNA, what is that? tRNA, what is that? And rRNA, what is that? There's some extra ones like microRNAs, siRNA, long non-coding RNAs. Those have different functions that you can memorize on your slides. But the next big concept is this difference between what the word polycistronic and monocistronic mRNA means. So both of these are mRNA. And there's a lot of differences between these. A lot of exam 3 is comparing and contrasting stuff. So polycistronic mRNA is in what organisms? Prokaryotes. It both starts with the letter P. Genes in polycistronic mRNA or polycistronic organisms are contiguous. Genes are contiguous, meaning the genes are right next to each other in the genome. There isn't large gaps of blank stuff, that the genes are very close to each other, very proximal. In fact, because they are very close to each other, a single mRNA that is made will contain multiple genes. So a single mRNA can make multiple proteins. All right. Let's contrast these features with monocistronic mRNA. Well, if polycistronic is prokaryotic, monocistronic is eukaryotic. Genes are not contiguous here. In fact, even in just one gene itself, it has things called introns and exons. The exons, if you don't remember, are your protein coding sequences. So let me write this as one gene will have these things. We'll have introns and exons. Exons are the protein coding stuff. Introns, I remember it as intervening. They're in the way and they need to be spliced out. So for eukaryotic mRNA, the way this works is when eukaryotic genes are transcribed, you actually have pre-mRNA, which still contains the introns and exons. But when you splice out the introns and you put together the exons, you have regular old mRNA. In this one copy of mRNA, you will only get one protein made. And that is where the phrase monocistronic comes from, is that one mRNA will only give you one protein at a time. Contrast that with polycistronic, which is in prokaryotes, where one mRNA gives you multiple genes and multiple proteins. All right? Let's see. Anything else? I know it's sort of in 29b, but there's some other modifications we do to eukaryotic mRNA. We attach something at the tail called what? A poly-A tail, good. And at the head, we attach a 5' guanosine cap. So those are some extra things that are associated with eukaryotic mRNA. And there you go. All right, yes?
And that's what causes HIV?
No. HIV, and I don't remember what section of notes that is, but HIV is caused by the fact that it mutates often, because it uses a reverse transcriptase, which makes mistakes all the time. That's the one I always remember off the top of my head. I can't remember the other two, but it usually is not associated specifically with this mechanism. Perhaps the mRNA, I know what you're talking about. The HIV, because it has a 5' G cap. It doesn't, I guess sort of friendly is like a good kind of analogy of it, but yeah, because it has a 5' G cap, it's like, yeah, it isn't a prokaryote. The cells don't know that it's harmful, okay? And yeah, that's the only thing with the HIV notes is that they're scattered all about between 10 to 29. You will get one or two HIV type questions where it's like, why is this so hard to treat? So you'll have to scan through your notes to find where that is. I may go over it tonight, so we'll see when we get to that. I believe after this is siRNA. I'm probably, again, skipping stuff like ribosomes or whatever, so feel free to review that. But after this, I usually remember is siRNA. What's that? Is the ribosomes? Right. So ribosomes, there's big ones and there's small ones. Eukaryotes have bigger ones than the prokaryotes, and there's some numbers associated with them. That's what I remember. So siRNA is a special type of RNA. This is sometimes your extra credit question, and if it's not your extra credit question, you will get multiple choice questions on it. siRNA is one of the many ways that we can perform RNA interference, or RNAi for RNA interference. That is the prevention of the expression of a particular gene. Because remember, in the central dogma, and oh man, I actually forgot to go over the central dogma, we have DNA, then you also have RNA, and then you also have protein, and it follows this pattern. DNA to RNA is transcription, we know that. And RNA to protein is translation, we know that. DNA has the ability to replicate, and there's this extra little thing that goes here called reverse transcription, of which some cells and some organisms are capable of doing. Okay. So, siRNA, the goal of siRNA is to inhibit this step, is to inhibit the translation step. The reason why is because the goal of siRNA is to make, well actually I'll go over the steps first and then we'll see the end result. How are you supposed to answer a question about the extra credit if it has to do with siRNA? Describe what its role is, describe how it's made, and the exact function in terms of how it actually prevents translation. And you need to give one applicable example of siRNA. I think the slides after this give you like 5, 6 different examples. Just tell me about one of them, and you'll be fine. So, here's how it works. siRNA originates from double-stranded RNA, of which there's 3 sources. I can't remember 2 of them. I know one of them is from RNA viruses, so feel free to fill in the other ones. They originate from double-stranded RNA. There is an enzyme called dicer that will dice up double-stranded RNA to create double-stranded siRNA. siRNA stands for small interfering RNA. Some people remember it as silencing RNA. That's fine. Either name is fine. It helps you remember what it does. But Dicer takes some double-stranded RNA, cuts it into little tiny double-stranded pieces. Each of those tiny pieces are double-stranded siRNA. And because they're so small, they will eventually unravel themselves till you have single-stranded siRNA. So that's how siRNA is made, is simply the enzyme Dicer cuts up double-stranded RNA. The siRNA by itself can't do anything, so the actual activity involved in siRNA is that it's going to combine with a whole bunch of proteins to form something called the Risc Complex, which there's an acronym for. I don't care about the acronym. You don't have to tell me the acronym. But this Risc Complex is going to take this small fragment of siRNA and try to match it to a target mRNA. All right, so mRNA can be really long, while the siRNA might be only a couple tens or 100 base pairs long. You have this whole mess of proteins that are holding on to this siRNA and looking for an mRNA strand that it matches to. And what I mean by match to is literally matching the bases to each other. If it finds a particular mRNA that it happens to match to, one of two things will happen. One, if the match is very good, if they are very complementary to each other, Risc will cut the mRNA into pieces. If you cut the mRNA into pieces, you can't correctly translate it into a protein because it's in pieces. Two, even if the siRNA is not perfectly complementary to the target mRNA, if it's good enough, the Risc complex will stay bound to the mRNA, thus preventing it from fitting into a ribosome and preventing it from being translated into a protein. The end result of either of these is you're preventing the production of a protein. Now, siRNA mechanisms happen naturally, but in terms of biomedical application, we can design specific siRNA patterns. We can design specific siRNA patterns, and we insert it into cells with these Risc enzymes, and they will go and they will find target mRNAs, harmful gene expression, and downregulate those things. All right, so that is one line of investigation associated with the siRNA. I don't remember what's next, so it's probably a good time to stop. I will go over more detailed stuff at the exam review tonight, and Thursday I'm going to have to start on chapter 30 stuff. But you see what I did? Try doing that for every chapter. Try to get as much out of memory as you can and practice doing that. Yeah. 29B will not be on exam 3. Yeah.
Because on the PowerPoint's like 29 part 1.
Oh yeah, I know 29. I know eukaryotic transcription is like at the very end of 29A, but you don't have to worry about that one either. Any eukaryotic transcription stuff, don't worry about it. Yeah. Yeah. So only the prokaryotic transcription is the last thing you need for exam 3. Yeah.
Josh,
Yep.
if you can,